My name is Mcelfish, and I'm a fifth-year PhD candidate at UC Santa Cruz in the Ecology and Evolutionary Biology Department, and I'm advised by Dr. Rita Maida. My work focuses on the nearshore fish community off Southern California's Santa Catalina Island. So, let's dive in. Under the waves, you can explore magnificent kelp forests, expanses of soft sand, and bouldery rocky reefs. Broadly, I seek to understand how different species collect and use sensory information. Why? Ocean conditions are rapidly changing. Increased turbidity from algal blooms or runoff directly impact fish who rely on sight, while warming waters can impact how chemical signals like odor cues move through the environment. But for many of these species, we currently have no idea what senses they rely on. My job is to try to figure this out. As you go through my poster, you'll see that I'm tracking behavior, measuring eye morphology, and looking at neural anatomy. For fieldwork, we travel to Santa Catalina, a small island off the coast of LA, where we work at the Wrigley Institute in Big Fisherman's Cove. For us, studying behavior is the extra fun part, because while kayaking or snorkeling, we deploy baited cameras to test sensitivity to odor cues. This video footage lets me predict species that may perceive their world heavily through smell. We got to sample two habitats also, sandy bottoms and rocky reefs. In the sand, we saw 3 species of benthic invertebrate grazers reliably enter the footage, including that pink fish that just swam by, a sheephead. We also had some special visitors, like this endangered 200-pound black sea bass. However, the checkered kelp bass, who is an active fish predator, were the most common visitors in the sand. In the rocky reefs, our kelp bass were still the most common, but they were reliably joined by my favorite, moray eels. For more information on the species we observed, check out the results on the poster. Okay, behavior? Check. Morphology is next. With REU students Gabby and Danny, we riffled through so many museum collections and spent many hours measuring fish eyeballs. Preliminary results on our poster show some clustering by foraging mode, with species that pick invertebrates, brown boxes, having larger eye structures. But our moray shows the smallest visual structures all the way on the left in gray. The poster breaks this down more, so be sure to check it out. Last but not least, neural anatomy. Fish brains are really cool. They have specific regions for processing different types of sensory information. And my goal is to compare these sensory processing centers between species. This work is just kicking off, and I can pursue this project thanks to generous funding support from the ARCS Fellowship. So to summarize, by combining behavior, morphology, and neural anatomy, I'll be creating a sensory catalog for fishes within this region. And this gives us a predictive framework for understanding how environmental changes may impact sensory behaviors. Thanks for listening, and let me know if you have any questions.